It, it's great to be here and get a chance to talk to you all. Um, I've already forgotten how to pronounce your name. I'm really sorry.
Ion? Sorry.
Say it again.
Ion.
Ion. Yeah. So
Yeah.
as Ion was was saying, our lab is interested in trying to understand how the brain generates complex sequential behaviors, and much of what we humans do is an example of complex learned sequential behaviors, like speech and language and music and athletic performance. These are all things that our brains have to generate a precise sequence of motor gestures. Those gestures have to be produced with the right timing and in the right temporal order for any of those behaviors to be successful. And so we try to understand how the brain generates and learns those things. We study the songbird as a model system. Songbirds are really cool because like humans, songbirds learn to vocalize by listening to their parents. So they go through a phase where they listen, they they form a memory, and then they try to imitate the parent's song. And so just like humans, we learn by imitation, and in most cases we learn those things through a huge amount of practice, right? So it takes us years to learn how to speak or to play an instrument or those things. And similarly, it takes birds a long time to learn how to sing. so we study the zebra finch as a model organism. Zebra finches are originally native of Australia, but you can see them, they're quite common in pet stores, so you can see zebra finches in pretty much any pet store. The singing is a sexually dimorphic behavior. So in zebra finches, like most songbirds, it's the male that sings. And it's basically a courtship behavior. So they sing to a female and the female chooses which male she wants to mate with based on the song that she hears. So the zebrafinch song sounds like this. Each zebrafinch has its own kind of unique song, which you can hear. So here's one example. Okay, can you all hear that? Good. Yeah. And here's a different male zebrafinch. Okay, and here's yet another one. I like that. That's my favorite one. He's very jazzy. So, so zebrafinch song has a very characteristic structure. They sing a bout of song, which is what each one of those recordings is. Each bout of song has a song motif that lasts about a second. So, and that motif is repeated over and over again. Each motif comprises 3 to 7 syllables. So each one of these little bursts of sound here is a song syllable. And those last about 100 milliseconds long. And between each syllable is a little 30 millisecond gap where the bird is actually taking a little breath so that he can keep singing for many seconds. Okay, pretty cool. Yeah, and just to remind you, this is what's called a spectrogram. So this is frequency on this axis. This is time on this axis. And each moment in the song, you basically compute a little Fourier transform of the pressure waveform, and that gives you a representation of how much power there is at each different frequency at each different moment in the song. Okay, so that's a song spectrogram, and that's the structure of the zebrafinch song. So as I mentioned, zebrafinches learn their song. They do that through a series of stages, just like humans learn to sing, learn to speak. This is a picture of a juvenile zebrafinch. Here you can identify them because they have black beaks. When they reach sexual maturity, their beaks turn bright orange. And in the background here, you can see a male zebrafinch. And what usually happens is the male sings to the juvenile, and the juvenile forms a memory of what the male sings. And that happens during a period of, around a month or 2 months after the bird hatches. Okay, so they're quite young at that stage. And then they begin to babble. In this sensory motor learning stage, they begin to babble and produce highly variable sounds. And then there's vocalizations. They gradually learn to imitate what what the adult sang. So here's what that progression looks like. The parent song is called the tutor song. And so I'm going to show you a tutor song down here. And then the stages of vocalizations this particular juvenile bird made as it goes from babbling to an imitation. Okay, so here's the tutor song. Okay, now around the time the young bird hears the tutor song, he begins to babble. Okay, so that's a baby babbling bird. A few days to a week later, you start seeing this kind of repetition in the song. So subsong, that babbling, is highly variable. The individual syllables have a highly variable duration. But then what happens is the bird starts having repeatable rhythmic syllables. So listen for that rhythm in this song at the next stage. Okay, so you can hear that kind of rhythmic pattern at about 10 hertz in the song. Then a few days later, the bird starts getting repeatable elements. So here you can see there's this particular sound right here that gets repeated over and over again. And this particular sound, let's say right here, that gets repeated over and over again. Okay, so listen for that. Okay, and then a few weeks after that, the bird reaches a mature song. And here's the adult song of this young bird. Okay, and to remind you what the tutor sounded like. Okay, so pretty good, better than I could have done. All right, so now, so what's really important here, though, is that these young birds have to hear a tutor song in order to develop a normal song. Okay, if they don't hear a tutor at this early stage here, they still develop a song, but it's highly abnormal. Okay, so here's an example of an abnormal zebra finch song that developed in a young bird, in a bird that never heard a tutor. And this is called isolate song. Okay, so you can see that that song doesn't have any of the normal motif and syllable structure of a normal zebra finch song. Okay, any questions about that?
I'm actually wondering if you could expound a little bit on what you mean by tutor. Is this you consciously exposing these birds to this, or are the birds consciously tutoring the littler birds?
Yeah, so so in the wild, the adult birds and the juveniles sort of all live together in a colony. In the lab, we breed the birds ourselves. You put a male and a female in a cage. They build a nest. Female lays eggs. The eggs hatch, and then at about 30 days of age, the young birds fledge, and they're able to take care of themselves. And about that time, the male is basically just constantly singing. So male zebra finches like to sing a lot, and the juveniles hear that, and that's the tutoring process. Thank you. Great question.
Quick question.
Okay.
Sorry.
Yep.
So the graphs that you have there, what are they indicative of? Are they indicative of like magnitudes or volumes at specific frequencies?
Yeah, exactly.
Okay.
So each one of these is a song spectrogram. And it's the same format that I showed back here. It's a different color scheme here, but the brightness of these, of each pixel indicates how much power there is at that frequency, let's say 7 kilohertz, at that particular moment in the song. And then 100 milliseconds later, you can see that there is a lot of power. First of all, it's more spread out here, but there's a lot of power down here at 3 kilohertz at that moment in the song. Okay, right. So song spectrogram is a time, it represents how much power at different frequencies at different times. Okay.
What kind of machinery or like thing you use to record like the sounds of the songbirds?
Yeah, so it's very simple. Basically, we take a bird, we put it in a soundproof chamber, and that soundproof chamber has a microphone inside of it. The microphone connects to a little amplifier, so it's just like a web microphone, that you plug into your computer, and then that's digitized, by a computer and stored in a file.
I have a question.
Mm-hmm.
If the juveniles need to learn the song from the tutors, how do the tutors learn the songs?
It's turtles all the way down, right? The tutors learn the song by listening to their own father, let's say, and that one learned from another bird. So songbirds have a song culture. They basically pass on from generation to generation. Okay, so mostly what we do in our lab is try to understand the brain circuitry that's involved in producing that song and in learning that song. Okay, so I'll show you some of the ideas that we think about in terms of how that actually happens mechanistically at a circuit level. And I'll show you some of the experiments that we have done and others and my colleagues in the field have done to sort of test these hypotheses about how this system works. Okay. So the basic formula, the basic idea for how vocal learning works in songbirds comes from Kenji Doya and Terry Sinowski, who proposed the notion that songbirds learn their vocalizations by reinforcement learning back in the 80s. And the basic idea of reinforcement learning is this. So there's some circuit in the brain called the song motor system that generates a vocalization, okay? And each time the bird sings, that vocalization is a little bit different. And that is produced by, you can imagine, like a variability generator, like in the brain that makes the motor output, that makes the control of the vocal organ a little bit different each time the bird sings, okay? So each time the bird sings, there's some different song that comes out. Now, the idea is that what's happening is that each time the bird sings, the bird is comparing its own auditory feedback of what it sings. So it hears itself sing. And that auditory feedback goes to a part of the brain that compares what the bird just sang with the memory of the tutor song. Okay. And then that comparison yields a signal called an error signal or a reinforcement signal that says whether that particular song was better or worse than past performance, let's say average of the previous songs. Okay, so now what can happen is that the motor system or the variability generator can start making more often the good copies of the song, the good versions of the song that best match the auditory memory. Does that make sense? All right, so that's reinforcement learning. And it's a basic conceptual model for how we learn a lot of things, right? Like many things that we learn, like a musical instrument, we make a lot of mistakes, right? And then somehow our brains learn to do the things in the way that don't lead to a mistake, right? Okay. All right, so the idea is that then after some period of time of this variation, comparison, reinforcement, that eventually the song motor system produces a good copy of the memorized song. Okay.
Another question, Dr. Fee.
Yep.
So when it comes to, let's say, language, right? What we're looking to correct when, let's say, I guess unconsciously, what a baby's looking to correct if they babble and trying to form words would be, the kind of word they're forming, the intonation with which they speak, things like that, right? What I guess I'm not sure if this is cataloged, but is there a kind of record of what a bird is aiming to correct, as in like the standards of its language that it's trying to match accurately to
Yeah.
whatever
So
memory it's looking at?
yeah, I would say we don't really know. It sounds to me like your question is kind of, how does this thing work? Like, what is important in this comparison process? And the truth is, this little box right here is at this point the least understood part of the system. Okay, so I would say we have very little understanding of how to answer that question right now. But I'll tell you what I'm going to tell you about today is this is this thing. So I'll start by telling you our sort of best guess as to how this song motor system works and how the variability generator first of all, that there is actually a variability generator that causes variations in the song and sends feedback to a part of the brain that can actually modify or improve the output. Okay. All right. So let's start with this. I'll describe the basic mechanism that we think is at work in the circuit that generates the song, and that I'll show you that circuit makes sequences that are then used to drive the vocal output. And then I'll talk about a neural circuit that implements reinforcement learning. Okay. All right. So let's talk about the brain circuit that generates the song. So the zebrafinch brain has a bunch of brain areas in it that have been identified as important for song production. Most of that work was actually done in Fernanda Notenbaum's lab at Rockefeller University. And what they did was they identified a bunch of brain areas in the songbird brain that are important for learning. They are for song production. They actually discovered that by studying the canary, because canaries sing seasonally. They sing in the spring during the mating season, and then in the off season, they don't sing. And it turns out that what they discovered is that some parts of the bird's brain are big in the spring and essentially disappear in the off season. They get much smaller. And so they had this idea that, well, those things might be related to singing. And then what they did was they kind of did a bunch of experiments to work their way back from the vocal organ up into the brain. And they discovered this set of brain areas right here, that when you lesion them or inactivate them, the bird is unable to sing. Okay. So this is the song motor circuit or song production circuit. The vocal organ is down here. The vocal organ is a lot like our vocal organ, but our vocal organ is up at the top of our trachea. Okay. The bird's vocal organ is at the bottom of the trachea at the point where the two bronchial tubes join. And there's actually, for us, in our vocal organ, there's actually one slit that air passes through and it vibrates when air goes through it. In birds, there's actually two of those, one on each bronchial tube. So birds can actually sing in two voices at the same time, which is pretty cool. the muscles of the vocal organ control the tension and mass distribution on this vibrating piece of tissue. Those muscles are innervated by motor neurons in the brainstem, just like motor neurons in our brainstem and spinal cord that control speech and movement. Those motor neurons get input from several brain areas that are in the analogous, they're analogous to cortex in mammals. So those brain areas are in a part of the avian brain that's thought to be very similar to cortex in mammals. Okay, and so there's this. This is sort of the very simplified cartoon version of this circuit. It's actually a little bit more complicated than that. There's a loop here where this output of cortex, this area called RA, it actually has a more complicated Latin name that I'll spare you. And that output of RA goes down to motor neurons, but it also goes to this area called DM. DM is interesting because DM is actually a part of the brain that's important for innate vocalizations in non-songbirds. So I should have explained this in a little bit more detail, but birds, about half of bird species are what are known as songbirds that actually learn their vocalizations by imitating their parents. The other half of birds are non-songbirds that just make innate sounds. And in non-songbirds have this area DM that's responsible for making all of those innate sounds. But songbirds have this whole complex structure on top of that that's important for vocal learning. Okay, so it's pretty cool. It's like they have the basic module, and then they have the advanced circuit built on top of that evolutionarily. Pretty neat.
Good question. Sorry, I keep interrupting.
No, it's okay.
Is the RA analogous to like the primary motor cortex in humans? Is it?
Yep. So
Okay.
RA is, let me be, that's great guess.
Okay.
It's, I would say it's a little bit more subtle than that. RA is kind of like layer 5 neurons in primary motor cortex. Okay, because the avian brain so RA is in a part of the avian brain that's like layer 5 of cortex. Okay, so the avian brain doesn't have this layered structure, but instead all of layer 5 is kind of lumped into one area back here. Layer 4 is kind of distributed into a few little lumps, and layer 2, 3 is some other piece of this thing. So it's not organized exactly the same way as mammalian cortex, but it's analogous. Okay. All right. Now, there's also a completely separate circuit that's important for learning, but not song production. So this circuit that I just showed you here in red is, if you lesion any of those brain areas, the bird can't sing anymore. But there's this whole separate circuit here. I don't know if you can see this. I thought this was in a different color than white on white background. But you can see this circuit here is basically analogous to a basal ganglia circuit in the mammalian brain. Okay, so songbirds have a basal ganglia, and within cortex is a cortical basal ganglia thalamocortical loop that's just like basal ganglia cortical thalamocortical loops in the human brain that are important for things like motor learning. And what's interesting is you can lesion any of these areas here in an adult bird that's already learned its song, and there's no effect on singing. But if you lesion those areas in a juvenile bird, they can't learn their song anymore. Okay, so it's kind of cool. So the way I think about this is this circuit is responsible for producing the adult song, but this circuit is responsible for programming up the motor circuit. Okay, pretty neat. Okay, and so there are very interesting analogies. So here's an example, a figure from Eric Jarvis's lab at Rockefeller. You can see in green is the human cortex. In green here is the analogous region in the songbird brain, okay? Here in pink is basal ganglia, and here in pink is basal ganglia in the bird, all right? And Eric Jarvis has even gone so far as to propose that there are very specific regions in the human brain, in the human cortex and basal ganglia that are analogous to these song regions in the songbird. Okay. All right, so let me start showing you some experiments that we've done to figure out how songbirds actually sing. So one of the first things we knew that we wanted to do was to record from neurons in birds while they're singing, okay? Now, it's kind of tricky because zebra finches are very small. It's a mating behavior. Most of the time when you want to record, like for example, in a monkey, in a behaving monkey, you put a plate or a pin on the monkey's head and you can head fix them and they'll still play a video game while you're recording neurons in their brain. You can't do that with a zebra finch. So you have to build something called a microdrive, which goes on their head. Zebra finches don't like to be handled. So most of the time when you want to adjust an electrode on a microdrive, you have to pick the animal up and turn a screw. That doesn't work on zebra finches because again, it's a mating behavior and that just disturbs them. So we invented this little microdrive that's got these tiny little motors on it that allow us to advance the electrode in and out of the brain by remote control. Okay. And so then what you do is you surgically implant this onto the skull under anesthesia. You put the electrodes down into the brain, and now you can advance these electrodes in different regions that you're interested in recording. Okay, so let me show you an example of some neural activity. So this is an example of a recording of a neuron during singing in this particular area called HVC. And these neurons that I'm showing you have been identified using a special technique that allows you to find neurons, for example, in HVC that project to RA. So there are a bunch of different kinds of neurons in HVC, but we were specifically interested in the ones that project to this motor system. So we used this technique called antidromic identification that allowed us to find those neurons. We recorded them during singing, and what we found is these neurons do the most amazing thing. They generate a single burst of spikes at a single moment during each one of these song motifs. Okay, so here's that burst occurred during that syllable during motif 1. Here's another burst during the same syllable in motif 2, another burst in that same syllable in the next motif. Okay, each one of those bursts is very precisely reproduced. Okay, so now we can record from a whole bunch of those neurons in the same bird or in different birds, or sorry, in the same bird. And using the song as a timing reference, because the songs are so precisely reproduced, we can line up all that neural activity to the song and see what that whole population of neurons is doing. So you can see that each one of these, so each one of these dots here is one of those spikes right there during a single repetition of the song. So this shows the spikes during 10 different repetitions of the song. And each one of these blocks of color shows a different neuron. Does that make sense? So you can see that this neuron generates a spike at the beginning of this syllable. This neuron makes a burst of spikes at this point in that syllable. This one makes a burst at that point. This makes a funny looking burst at that point in the next syllable, and so on. And each one of those neurons only makes a single burst during the entire song. Okay. And if you record from a much larger number of neurons, which we were able to do years after this first publication in 2002, we found that if you record from a lot of those neurons, they completely tile time in the song. In other words, every moment in the song is represented by one of those neurons. Okay. Now, based on the number of that type of neurons in HVC, we think that each moment, there are 20,000 of those neurons. Each one is active for, let's say, 5 milliseconds out of a 500 millisecond song. So each one is active for 1% of the duration of the song. And if there are 20,000 neurons, then there should be 200 of those neurons active at each moment. Okay. Does that make sense? Okay. Now, let's go down to RA. So that was HVC. Now let's look at RA. So RA neurons do something quite different. So they also generate bursts during the song, but they generate a pattern of about 10 to 12 bursts during each repetition of the song. Okay. And again, those bursts are still very precisely reproduced. But when you line up the activity of a whole bunch of different neurons, so each block of color here is a different neuron. If you line up the activity of all those different neurons, you can see that it's a much denser pattern of activity during singing. Now remember, these neurons here project directly down to motor neurons that control the muscles. Okay. All right, so how might this actually work? So our hypothesis is that these neurons in HVC sort of form a representation of time. Each one of them represents a single moment in the song. And each moment in the song is represented by a different group of neurons. Okay, like that. And the idea is that those neurons activate downstream in RA some population of RA neurons, okay, like that. And those RA neurons converge onto the motor neurons in the brainstem or spinal cord. And that produces, those motor neurons then activate the muscles to produce some pattern of tension in those muscles that evolves over time. Okay, so it's a very almost mechanical music box kind of view of how it works, right? You can almost think of this HVC as like the timer or the thing that rotates the drum on a player piano, and these connections from HVC to RA are like the holes in the paper on a player piano, and the RA neurons are like responding, you know, they're activated by the holes in the paper, right? So it's like almost this music box view of how it works. Any questions about that?
So multiple neurons in the multiple neurons in the HVC can innervate a single neuron in the RA, correct?
Yeah, so okay, let me fill in a little bit more of the numbers.
Okay.
So there are 20,000 of these neurons. We think that corresponds to, if they're evenly distributed in time, which it seems they're approximately, that means there are 200 neurons coactive here. There are 6,000 or so neurons in RA, okay? And at any given moment, about 10% of those is active. So about 600. So what we think is that at any given moment, there are 200 neurons in HVC that are activating about 600 neurons in RA.
So when you increase the amount of neurons you're measuring in the HVC, was there an increase in the resolution of, not resolution, sorry, was there like a decrease in the space between each neuron firing? So when you increase from 10 neurons, or I think it was 7 or 8 neurons to studying, yeah. Okay.
Exactly. So here you can see, if you sample a small number of those neurons, you can see there's a lot of gaps in here between them. And that led to actually a lot of speculation in the field that maybe those gaps are really there. Like maybe HBC isn't doing anything during those gaps here or here. Okay, and that's why we went and actually recorded from many of these neurons to show that those neurons actually completely cover the song. Okay. Now you say, okay, what about this little gap right here? Or what about this little gap right here? But if you do a statistical analysis on the distribution of those bursts in the song, it's exactly consistent with random placement of those bursts.
Could
So
you
we
elaborate
were-
on that? Sorry, I didn't quite
Yeah,
understand.
well, I mean, basically the point of this paper, this neuron paper from 2016, the point of that paper was to show that those theories are wrong, that the theories that there are big gaps in the song, in the HVC activity, we did those experiments to show that those theories are wrong, that the activity in HVC is continuous in time. Okay, so now let's turn to the question of how those sequences in HVC are actually generated. Okay, like what controls the timing of those sequences? So one possibility is that those sequences in HVC are a result of circuitry within HVC. So you could imagine that that group of 200 HVC neurons is synaptically connected to the next group of 200 HVC neurons. And those are connected to the next group, and those are connected to the next group, right? In a like a chain of activity. Okay. Now there are different models for how you could get a sequence of activity in HVC, but the basic hypothesis, one basic hypothesis, is that the circuit that produces that sequence is inside of HVC. Okay. Now there's an alternative circuit. Sorry, I'm just going to jump ahead to the alternative circuit. The alternative circuit is that those sequences in HVC are directly driven by circuitry somewhere else. Let's say in that thalamic nucleus that I barely mentioned, okay? All right, does that make sense what those two different ideas are? The one is, so the one idea is that the circuitry that generates that timing, that generates that sequence is within HVC, and the other possibility is that it's outside or upstream of HVC. Okay. All right, so I'm going to show you an experiment that we did to test those two different hypotheses. Okay, to basically rule one of them out. Okay, all right, so here's how it goes. So let's imagine an experiment in which you could slow down the circuitry inside of HVC. Okay. Now, under this hypothesis, if you were to slow down this circuit within HVC, you could imagine that it takes longer for these neurons to activate those neurons, and it takes longer for these neurons to activate those neurons, and so on. So you can see that the interval between the onset of this burst and the onset of the next burst would increase, right? And therefore, there would be fewer of those bursts per unit time, and the song would slow down. Okay. Now, on the other hand, under this hypothesis here, you can see that if you were to slow down the circuitry inside of HVC, it might take a little bit longer for this neuron to activate this neuron, and it would take a little bit longer for this neuron to activate that neuron. But that delay would be exactly the same, and the song would not slow down. So in other words, if UVA was the clock, UVA activates HVC at some regular interval. slowing down HVC would add a little gap there and a little gap there, but that gap would be the same everywhere, and you would get exactly the same number of bursts per unit time. And so the song speed would stay the same. Okay, so how might you slow down a neural circuit? Does anyone, maybe somebody read the paper, or somebody might want to just guess.
You could use hold on, you could use like some kind of like a hyperpolarizing drug or something that's like a GABAergic agonist or something, and try to
Yeah.
prevent
Okay,
neurons from
Yeah.
from firing.
Yeah. That would probably do it. We never tried that. We tried a different approach. We used temperature. So you may all know that that neurons are highly temperature sensitive. That's why fevers are so bad for the brain. They cause hyperexcitability of neurons. And if you cool neurons, they become less excitable and slower, like all the synaptic processes slow down. Okay. So what we did was we built this little brain refrigerator. So these experiments were done by Mike Long, who's now at NYU when he was a postdoc in the lab. And we built this little brain refrigerator. It uses the Peltier effect. So you run current through it one way and it cools these little metal pads that are on the brain above HVC. And if you run current through it the other way, it heats the metal pad. Okay. And you turn the current on and you can see very rapid over 10 seconds, a change in the brain temperature, and you turn the current off and the temperature goes back to normal. Okay. All right, so here's what happens. Here's the control song spectrogram. So that's the normal song. If you heat up HVC, the song gets a little bit faster. You can't heat it up too much, otherwise you lesion HVC. But if you cool it, you see that the song gets longer and longer. Okay. And now let's take a look at kind of in detail how the song changes. So you can take, so here's a spectrogram of the normal zebra finch song. Here's the spectrogram of the cooled song. And if you take a copy of that image in PowerPoint, grab the little handle and stretch it out, you can see that it looks exactly the same as the cooled song. Okay, so what's happening is that the whole song is just being stretched out as if you're just turning down the speed of the clock. Okay. And you can quantify exactly how much the dilation of the song depends on the temperature change in HVC, and the slope of that line is about 3% per degree, of 3% change in the speed of the song per degree C of temperature change in HVC. Okay. All right. So, okay. So our favorite hypothesis is that these sequences in HVC are produced by a synaptically connected chain of neurons like this, where this group of 200 neurons activates another group, and so on. There's a slight subtle variation of this model, where what we think is actually going on is not that the whole sequence is driven by a chain that exists within HVC. What we think is that each syllable is essentially a chain in HVC, and that the end of one syllable activates the beginning of the next syllable through this loop that I described before. Okay, so the idea is that the last neuron in syllable A will activate through this loop the first neurons in syllable B. Last neurons in syllable B will activate the first neurons in syllable C, and so on. Okay. Yes, question.
Yeah, could you just reiterate what initiates the first sequence in the HBC?
Oh, this first neuron right here.
Right.
Yeah, well, so we don't really know. We suspect it's probably uva, but but zebrafinch song actually has an interesting structure. Zebrafinch song is often initiated by what are called introductory notes. These are just little bursts of vocalization that also are active at like a 10 hertz rhythm. And we think that that may be kind of involved in sort of kickstarting the system to start singing. Okay, but we don't really know the answer to that question. All right, any other questions? Okay, so we've had a luxurious, slow walk through the song production system, which is great. You've asked a bunch of really awesome questions. I'm going to give you a much abbreviated picture of the song learning circuitry and a few of the experiments that we have done to kind of figure out how that works. Okay, so remember the song learning circuit, and again, the basic idea, which was actually proposed by Sarah Botcher back in the 80s, is that LMAN, which is the output of this cortical basal ganglia thalamocortical loop, basically sends an instructive signal that programs up the motor pathway. Okay, now what we have found since then is that the output of LMAN is not so much a set of simple instructions that programs the motor circuit. It's actually primarily a variability generator, okay? And the idea is that LMAN injects variations and injects variable activity into the motor pathway. And what the rest of this circuit does is essentially watches what LMAN is doing, like it gets a copy of what LMAN is doing in the basal, and that copy of LMAN commands gets sent to the basal ganglia. And the basic idea is that what the basal ganglia is doing is it figures out whether each one of those LMAN commands is making the song better or worse, and then reactivates the commands again that made the song better. Okay, so that's the basic idea. Let me just show you some of those experiments. But again, the idea is that HVC is a sequence generator that projects the RA that makes the adult song. LMAN is a variability generator that drives variations in the motor pathway. Okay, so let me just show you an example. So LMAN, we've shown LMAN actually drives the subsong babbling at the very earliest stage. Okay, so here's an example of the song, the babbling song, with LMAN intact. If you inactivate LMAN with bilesioning it or injecting a drug that inactivates the neurons, what you can see is all that variability goes away and the bird makes one exactly replicated syllable over and over and over again. Okay. All right. Here's just another example. So if you inactivate LMAN, you can see that here's an example of a song with normal LMAN, with an intact LMAN. You can see these harmonic stacks here, which I've copied up here, are highly variable. When you inactivate LMAN, you can see that those harmonic stacks become very, very clean, very stereotyped. And I've copied those up here, and you can see these are highly stereotyped syllables. And that's produced simply by inactivating LMAN. So we think LMAN is actually the variability generator in the brain that allows the bird to make different variations of its song while it's learning. Okay. I kind of think it's fun to think about LMAN almost like a creativity center for vocal learning, right? It's forcing the young bird's brain to try out different things as it's learning. Okay. Okay. All right. So let's see. Where am I here? All right. So basically, I have 4 minutes left. So let me. Okay. So the idea here is that L-man generates variations that try out different things in the song. Area X monitors those variations, figures out which ones make the song better, which ones make the song worse, and reactivates the variations that make the song better. Okay, so let me just show you an example of error signals in the brain. So how that process actually, how that part of the brain actually gets input about what makes the song better. So here's a very cool experiment that was done by Vikram Gadekar in Jesse Goldberg's lab. And so what they did was they recorded from neurons in the ventral tegmental area, VTA. These are dopaminergic neurons. Have any of you heard about the role of dopamine in reward in the brain? So dopamine is thought to be an important signal for driving learning. And the idea is that neurons in the ventral tegmental area, VTA, are activated by unexpected rewards. So there's a classic series of experiments from Wolfram Schultz's lab showing that if you record from VTA neurons and give a monkey an unexpected juice reward, that those VTA neurons become activated. Okay. Now, if you withhold a reward that the monkey was expecting, those dopamine neurons actually transiently become suppressed. Okay. So the dopamine neurons have been hypothesized to play an important role in learning by being activated when an unexpected reward happens and by being suppressed when an expected reward doesn't happen. Okay, and you can see that that kind of signal is really perfect for reinforcement learning, right? Where you need a signal that says whether what you just did gives you an unexpectedly good outcome or whether what you just did makes the outcome worse. And so we hypothesized that these VTA neurons that project to area X might actually carry a signal about song performance, okay? And Jesse Goldberg and his postdoc were able to actually carry out this experiment. They recorded from VTA neurons that project to X using the same technique of antidromic identification. Then what they did was while the bird is singing, they targeted a particular syllable in the song, and half the time they played a noise burst on top of the bird while he's singing. So here's the song with a noise burst, and here's the song without a noise burst. And what they found was that, that these VTA neurons, when the noise burst was present, exhibited a suppression of their activity. So here you can see all these spikes going along, da, 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 and then the noise burst happens, and then the dopamine neurons shuts off for 100 milliseconds. And then it starts spiking again. And on the trials, this is the coolest thing, and on the trials where there was no noise burst, that neuron generates extra spikes.
So if you were to study the bird song being generated after repeated trials of purposefully putting noise bursts at a particular pattern, is that part of the bird song then suppressed abnormally compared to like a control, or like a where there's no noise burst?
Yeah. So you're asking if you were to kind of play a noise burst on top of the song in a juvenile bird for many months, would you screw up the song at that time?
Yeah, exactly.
I am not actually sure. I think it's possible. It's also possible that the bird could actually learn its song on the trials where there's no noise burst. And that's actually my guess as to what would happen. Okay, but notice now that area X has a signal that says whether the song was better or worse than previous, like the average of previous trials, okay? All right, and this is just a reminder of the Schultz results showing that here's a VTA neuron when a monkey gets a juice drop that wasn't expected. And here's the neuron showing a suppression of activity when the monkey didn't get a juice drop that was expected. Okay. All right. So here's our sort of big picture view of how this works. HVC makes a sequence. In the normal adult bird, remember, HVC makes a sequence that controls RA neurons that then drives motor neurons to produce song. The way we think the young birds actually learn its song is that what it's trying to do is the young bird has a HVC sequence, but it doesn't have these connections from HVC to RA set up correctly. So LMAN drives variations in the RA neurons. Some of those variations make the song better, some make it worse. A copy of that variability command goes to area X. Area X also receives a dopaminergic signal that says whether that variation made the song better or worse. Area X can now figure out which variations made the song better and which made it worse, but it also has to do that calculation independently at every moment in the song. Because a particular variation at time 10 might make the song better, but that same variation at time slot 11 might make the song worse, right? So then the idea is that area X can then reactivate, using that timing signal, the same variation through that basal ganglia thalamocortical loop that made the song better. And that becomes biased variability, and that reactivates the good variations in the motor system. Okay, so that's, and then the idea is that those variations get permanently built in to those synaptic connections from HVC to RA. So if you imagine that LMAN discovered that activating this particular neuron at time point 10, sorry, at time point 3, makes the song better, then that synaptic connection from HVC neuron 3 to that RA neuron will get stronger through just Hebbian learning. Okay. All right. So that is our hypothesis for how it works. And I'm going to stop there.
Can I ask you a question?
Of course.
Okay, that was great. I really enjoyed that. I think I got a lot out of it. I have a sort of a weird question, and just feel free to put a break. So it seems like to me that these birds as a species, their brains have evolved to, well, the metaphor that's coming to my mind, it's like they're all getting very, very good at, say, like cubism, like one style of art. But, you know, it seems like that's what the LMAN is sort of doing. It's all driving towards getting good at doing this one form of birdsong, whereas, you know, I think especially in you know, the human species, we see a lot more variability, and we get a lot more out of variability. So I'm just wondering if you've ever done anything or thought about why they drive towards only one form of this birdsong.
So let me just give you a very specific example about human behavior where I think this is a really good model. So humans, it's true, do many different things. They learn many different things. You know, from, you know, different styles of painting, maybe to musical instruments, to speech, to athletic performance. Like some particularly talented person could even do all 4 of those things, okay? But the way that I think about this is that this particular circuit implements a very important behavior for the bird, And I think about it very much the way humans learn to speak. So, our speech and language learning system drives toward learning a particular language, a particular manner of speaking, a particular accent. Um, we have a remarkable degree of homogeneity in the way we use language in our own little group, right? Now, another group of humans learns a different language, a different accent, a different vocabulary, a different grammar, but they are all, through their culture, driving toward the same common language, right? So that's the way I think about it. It's language, the language learning circuitry in the human brain is highly specialized to produce one particular behavior that we imitate. And if we can't imitate the language of our peers, we won't be successful at, many things.
Thank you.
Sorry, I'm
Great
just gonna
question. Yeah.
I'm gonna ask one more question. If no one else has a question, I just wanted to ask. So you mentioned how the learning circuit is less influential in adult songbirds. So does that imply that um, as a songbird gets older, its song is harder to modify, or it's just that um, that the thalamic basal ganglia is it LMAT? Is that
Oh, man.
the
Yep. Yep.
yeah, that circuit just becomes so good at defining what a good amount of variance is, or it's so used to firing that particular pattern of variance that it just stays with that.
Yeah.
What
Yeah.
is it?
So basically, songbirds have critical periods just like humans. They have to hear their tutor sing sometime between the ages of 30 days post-hatch to 65 days post-hatch. Otherwise, they have a very hard time learning their song. They develop an isolate song after that age.
It's like a critical period. Like,
It's exactly a critical period.
okay.
So, and there's sort of lots of interesting subtleties and complexities to these critical periods. So if you, for example, if you take a young bird and you prevent it from hearing itself sing its own song, its song will degrade very rapidly. But if you take an older adult bird and you do the same manipulation, their song is stable for years. So there's all kinds of different ways that the juvenile bird has a lot of plasticity in its brain, but the older birds don't.
Cool. All right. Well, we're 10 minutes over. I'm sorry for
No
going over a
problem.
bit.
It was
But,
really great pleasure to talk to you guys and get your great questions. Thank you.
Thank you so much. Your presentation was incredible. It's very easy to understand something that's very difficult, which is a running theme, it seems to be, with our speakers. So that's really
Cool.
good. Thank you so
Good.
much.
Good.
Really appreciate it.
Okay. Yeah. Take care.
Take care, Dr. Fee. Thank you.
Yeah.
For the rest of you guys, we're actually having bye. We're having a movie night in a couple of weeks, and we're going to be having people come into the Rucker Cinema at Livingston and be watching the new Halloween movie, The Halloween Kills. And then after that, we'll be doing an analysis of Michael Myers, the character, with Dr. Tobiah, who teaches psychiatry at RWJMS. So because the theater itself has a limited capacity, we're sending out a sign-in sheet or sign-up form for people who are interested. So if you are interested, we'll be posting that sign-up sheet in the chat, but I'll also be posting it in the Discord. We'll be posting it in the Discord and emailing it out later. So just give me 1 second while I get that ready. Hold on. Who is interested in that right now? If you could give me a show of hands on Zoom or unmute and just tell me.
I have the link. You want me to send it?
Okay, yeah.
Okay. That might help. Here you go.
That one doesn't work. Try this. Okay, cool. Yeah, that's great. 4 of
Oh
you.
wait, is that the
Awesome.
wrong? Wait, that's the right
It
one.
might be the edit link. So if they can't
Oh,
access that
wait,
one, you
that
can
might
try,
be the
you can
edit
try
link.
you can try the bottom one. So awesome, guys. Well, you guys are free to go, but thank you so much for coming. It's you know, as always, it's late on a Wednesday night, right in the middle of the week, so we appreciate you coming, and hope you guys enjoyed. If you have any questions, you can just stick around for a bit if you want. But thank you, as always.
I just had a quick question about next week. Is it going to be online as well?
So next week we don't actually have a meeting, so we'll be sending an announcement out about that. We hope to just get as many people as we can to sign up for our movie night, and also it's midterm season, so we're trying to take a break for next week.
This is actually my first meeting. Is it possible to get signed on to your email list or something?
Yeah, sure. I have a link to the mailing list. So you can actually, I'll give you our link tree. So our link tree has all of our relevant links. So you can go and explore that as you wish. The mailing list is the second one down, but you can also join our Discord, and anytime you want to join our meetings, if we're having a meeting, our Zoom link is there if we're having a virtual component for it.
Okay.
Along with other stuff, yeah.
Yeah, have a good night.
Okay, yeah, you too, Ben. Thank you.